Great, thank you. Good afternoon, everyone. I'd like to thank the organizers for giving us the opportunity to talk today at lovely Barcelona. My name is Kevlahan. I'm the CEO and co-founder of Nanite, and I'm here to talk to you a little bit about what we're doing in non-viral delivery and our mission to deliver the undeliverable. So to introduce Nanite, we are a AI ML-centric organization. So we build bespoke non-viral delivery vehicles powered by an AI engine. And we really do it to understand the intersection of material science and biology. And our business model is quite straightforward. We partner with cargo companies and therapeutic companies to build bespoke non-viral delivery vehicles through therapeutic licensing and to develop best-in-class non-viral delivery. I'm joined with my co-founders, Shashi Murthy, Thomas Neenan, and Teresa Reinecke. And much of this work came out of Teresa's lab at the University of Minnesota with our 25 years of experience in designing polymeric-based gene delivery vehicles, as well as Felipe Oviedo, who stands up our machine learning team. But we brought together a team of polymer chemists, experimental biologists, and AI and computer scientists to really understand how to build next-generation non-viral delivery vehicles. So as everyone has been following the gene therapy market for the past decade, it's been really exciting to see the momentum, but there's still a lot of challenges that have yet to be ameliorated within the space. And you ask anyone in gene therapy what the biggest challenge is, and they will say delivery, delivery, and delivery. And so that is one of the things that has been plaguing the gene therapy market for quite some time. And it even gets even further more complex as now we brought on next generation non-viral delivery vehicles that have started to come into the limelight. So in delivery, it's a tale of two cities. There's the viral city and the non-viral city. In the viral world, which has been the flagship delivery mechanism to deliver for gene therapies, has been struggling with limited cargo size, difficult to manufacture. One of the most expensive drugs on the market today that's a gene therapy is a viral-mediated gene therapy. And which was interesting during the COVID era, and we developed the COVID-19 vaccine, the unsung hero that came out of that was the lipid nanoparticle. And that really cracked open a really interesting opportunity for transient-based therapies and really addressing some of the viral-based issues. But there's still some challenges in the lipid world as well, such as really difficult to detarget the liver, very complex landscape. And so we believe polymer nanoparticles are really the next generation non-viral delivery vehicle. It takes the features of lipid nanoparticles, but really opens up the chemical diversity quite a bit. And as well as having the ability to actually hang ligands off the surface of these polymer nanoparticles gives the ability to actually do affinity-based targeting. And it's a much different landscape in comparison to lipid nanoparticles. But the challenge that's been having within the polymer world is simply to ask the question is, where do you even start? And so as a material scientist, you end up in this really interesting space of chemical diversity. And this is really what Nanite is aiming to solve. So to give you a perspective on chemical diversity, in the polymer universe, you have the order of a chemical diversity of 10 to the 60. So the question lies, how do you even be able to dig or know where to dig within a 10 to the 60 design space? I'm going to give you kind of dollars and time here. Is that if you wanted to screen empirically a 10 to the 60 design space, it would probably take you 100 years and $1 trillion to do, right? It's just untenable to empirically determine everything and the intersection of material science to biology and seeing where polymer nanoparticles could fit within gene delivery. And so we thought to ourselves, so what if we were taking small samplings in this 10 to 60 design space and apply computation to predict biological performance? And that's what Nanite's solving. And to give you one of the big things that, in aspects of chemical diversity on the 10 to the 60, the amount of levers you have to pull within this space, whether it's average size of the polymers, the topography, whether they're linear, bottle brush, dendritic in nature, whether the monomer level is either a single monomer, is it multiple monomers, are they alternating, are they statistically co-blocked? monomers, and then balancing the charge type and hydrophobicity and hydrophilicity, and even to add ligands to the surface. You're going to just give you the order of magnitude on the things you have to optimize from a material science standpoint for non-viral based gene delivery. And so having to do a random walk process to figure out what actually is the best approach for a biological output without computation is really nearly impossible. So we asked the question, this is really where Nanite was founded on, we asked this one question, is that if we had a platform that we can actually be able to characterize stability, uptake, editing efficiency, tissue specificity, and toxicity, could we predict polymer architecture, monomers, compositions, and configurations that's driving that biological output? And this is the power of artificial intelligence and machine learning, is making inferences between a very large design space and actually being able to predict things, essentially a biological output. So we do this in two different ways. The first way we do is a wide search. And so we look at how polymeric-based nanoparticles applied into a biological system and understanding the global polymer attributes to the biological outputs. And then once we understand certain libraries localized to different organs within an in vivo system, then we do deep search for refinement. And so that's optimizing within a particular tissue type. So our wide search platform is called Sayer. And what we designed is a closed loop, high throughput process where we can predict, synthesize, complex, characterize, screen in vitro, and screen in vivo. And all that data is distilled and hashed to make predictive models to see what polymer-based chemical features are going where, what's driving endosomal escape, and and as well as what polymer features are actually driving certain complexation of different cargoes. So I'll walk you through a little bit on that workflow. So the first starts with our training data. So what training data is in the machine learning is basically you're training or teaching an algorithm on what polymer representations are, what charges are, and as well as how they interface with a biological system. So we take everything down to first principles. So we model cargo polymers from the molecular levels, as well as the interactions and how they form polymer nanoparticles. And then we take those polymer nanoparticles and apply them across different biological systems. And with that, we make billions of machine learning relationships that allows us to predict what chemical features are driving biological performance. And once we make a series of predictions, we generate a library of polymers. And so we're able to make 100 distinct polymer nanoparticle formulations in a day. And that's growing every day through some of the infrastructure that we built at Nanite. We can apply that polymer library across in vitro systems. Now, in vitro systems, tell one story, specifically around how well a polymer actually delivers and whether or not you get a baseline toxicity or viability. But we're able to take these whole libraries and generate on the order of 10 gigabytes of data per cycle. And we developed deep learning models that can actually process 10 gigabytes of data within 10 minutes. So that gives you a context of when you're applying machine learning and artificial intelligence, the generation and the throughput, and as well as how fast you process the data actually feeds very importantly within your machine learning models. And so that gives us a baseline of training data that we can actually make and understand what polymers complex with what particular cargo, how well do they actually perform as a baseline in a vitro system, and develop base case design rules, right? So as a biologist, one might think that applying one polymer nanoparticle across, let's say, a plasmid DNA versus an RNP should perform the same, but actually they perform vastly different and complex vastly different. So for instance, pointing your eyes to the center graph there, if you're looking at efficiency in this particular polymer library, if you wanted to build a polymer for complexation of plasmid DNA, you would actually build the polymer that is optimized for pKa. Versus if you're complexing with an RNP, you would look at Hill coefficient and zeta potential. Now, as a material scientist, these are very different polymeric-based properties. And so you would have to design these polymers very differently across the library. And so this slide is really where the rubber meets the road. This is where we get our largest corpus of data. And that's being able, and we're the first ones to do this, is taking polymer nanoparticles, being able to complex them, pool them into one singular sample, and bring them into one animal. And so we're able to effectively take multiple different polymer nanoparticle formulations and bring them into an in vivo system and extract essentially all the organs, and then being able to look at trafficking, tropism, and expression profiles and develop trafficking maps for each polymer nanoparticle that we formulate. And this really applies our low cycle time, low cost on actually generating a lot of data in a single clip. And so that brings us to one of our case studies. So this is a deep search refinement, right? So we apply these multiplexing experiments. We get a baseline. We see, in this instance, we're looking at the lung. We're seeing what is actually hitting within the lung. And then we take essentially the polymer family that starts to localize in the lung, and then we do closed loop optimization for targeted lung delivery. So you can see on the figure on the right, where you get starting off with, let's say, our worst case polymer that was localized in the lung. As we begin to constantly optimize on different within that polymer family, we're able to drive that about nearly five-fold, just through a couple cycles of closed loop iteration. So our business model is really to partner with therapeutic companies and cargo companies to develop in vivo non-viral gene delivery systems. And so we partner with various cargo companies ranging from those who have cargoes on the CRISPR-Cas9 systems to ASOs to mRNA to base editor. We're really cargo agnostic. So we build bespoke delivery vehicles across the gambit. We recently announced two research collaborations through some patient advocacy groups. The first being the Cystic Fibrosis Foundation, which we're aiming to deliver next generation polymer nanoparticles for mRNA delivery for cystic fibrosis. The next we just announced was the Charcot-Marie Tooth Disease Research Foundation, where we're delivering ASOs to Schwann cells to knock down PNP22. And the last and this is the last leg of our business, is our internal development pipeline. I mean, this is something that we're working pretty hard on prioritizing, and we'll probably be announcing in the coming year. So we're located in Boston, Massachusetts, specifically in the Seaport. We have a fantastic team at the intersection of polymer chemistry to biologists to computational scientists. And we're right next to the convention center, and we actually just opened up shop down in Boston. So with that, I think that's my last slide. I mean, happy to take any questions after the talk, and my email's up there, so feel free to shoot me a note. Thank you so much.